said that the audience has a very broad background. So to make up for this, I've chosen to talk about some very different, not to say disjoint topics, hoping that there will be something for everybody here. But please do not hesitate to interrupt and ask questions. My first example, assume that my slides will move, is taken from the field of theoretical ecology. This is the postdoctoral work of a former postdoc in my group, Yu Mong, in collaboration with my colleague Karl Modis in Dresden and his former postdoc, Szabolcs Horvat. And our starting point is the eternal question of theoretical ecology. Does an ecological community allow stable and feasible coexistence? To explain what I mean by this and to fix our mathematical ideas, as it were, let us assume that this kind of community is described by the Lotka-Volterra equations. X dot is equal to x times a minus b times x, where I'm using this slightly cavalier notation to denote that the abundances xi of each of n species evolve according to xi dot is equal to xi times some birth rates, ai, which can be positive or negative, minus some interactions, some of the bij xj, where again these interactions can be of either sign, but the diagonal terms bii are positive to denote some notion of carrying capacity. These equations have a single equilibrium of coexistence of all species, x star, which is b inverse times a. And I'm going to say that this is stable in the usual dynamical system sense if small perturbations away from this equilibrium decay back to it. So all the eigenvalues of the Jacobian at the steady state minus x star times b have negative real parts. I'm going to say that this is feasible if all the abundances at the equilibrium are positive. Now, of course, given ai and bij, it is very easy to decide, at least numerically, whether this equilibrium is stable and feasible, whether these conditions hold. However, ultimately, theoretical ecology seeks to describe the general principles that determine whether this equilibrium is stable and feasible. But in this context, there's a curious gap between systems with two species only, which is textbook material, and systems of very many species, which are usually described statistically using ideas from random matrix theory that were first introduced into theoretical ecology by Robert May half a century ago. But here we wanted to take a complementary approach and think about small ecological networks of just a few species. So we're going to think of these complete graphs, networks, with these Lotka-Volterra dynamics. There will be two types of nodes of corresponding growing species that grow in the absence of other interactions. AI is greater than 0, and species that die on their own. AI is less than 0, that require interspecies interactions to survive. Similarly, there are three types of edges: competitive interactions between species, bij, bji are positive; mutualistic interactions, bij, bji are negative; and directed predator-prey interactions. Species I eats species J if bij is less than 0 and bji is greater than 0. So together, these are going to define, for want of a better word, the network topology of the ecology. In what follows, I often want to make statistical statements. I'm going to sample ecologies of a fixed topology, so with fixed signs of the AI and bij, and sample the absolute values of the parameters AI and bij uniformly from a uniform distribution. I can then compute the probability that this equilibrium of coexistence is stable and feasible. In other words, I can compute the volume of parameter space in which stable and feasible coexistence is possible. Now, if you have a large sheet of paper, you will convince yourself that there are 10 ecological networks on 2 species. 4 of these are what we're going to call trivial ecologies. Because, for example, this ecology over here has 1 species that is dying on its own and only has deleterious competitive interactions with other species. So its abundance must decrease. There cannot be coexistence of both species. So stable and feasible coexistence is trivially impossible. The other 6 ecologies are non-trivial, but one of them is special. This obligate mutualism of 2 species is what we're going to term an impossible ecology, because stable and feasible coexistence is actually non-trivially impossible. Both species have negative and positive interactions. But if you write down the conditions of stability and feasibility for these dynamics, you will find a non-trivial contradiction. We can extend these ideas to 3 species. If you have a very large sheet of paper, you will convince yourself that there are 70 non-trivial ecologies on 3 species. And this swarm plot shows the probability of stable and feasible coexistence in each of them. This probability is 0 for 4 of these ecologies, which are the impossible ecologies on 3 species. Again, we can prove that they are impossible by writing down the conditions of stability and feasibility for these communities and obtain a non-trivial contradiction. There's another interesting ecology here. This one, which is possible, but the probability of feasible and stable coexistence of which is orders of magnitude below that of the other ecologies. And this has the interesting property that all its 2 species sub-ecologies are either trivial or impossible. So this is what we're going to call an irreducible ecology. And in fact, there are 6 irreducible ecologies amongst the 66 possible ecologies on 3 species. If we plot the cumulative distribution function of this probability distribution and restrict it to the irreducible ecologies, we see that it is reduced. Irreducible ecologies have a low probability of stable and feasible coexistence.
Yeah, yes. I asked this question the previous time I saw this talk, so I think I'm going to ask it again. So how do you define this probability?
So in this case, the probability is simply the volume of parameter space in which coexistence is stable and feasible coexistence is possible.
So you have all your coefficients of your 3 interacting species, which is something like 9 parameters. Or
yes, and then some growth rates. So yeah, it's a 12-dimensional space.
Yeah,
and there's a chunk in this parameter space in which coexistence is stable and feasible. So we compute the volume of this parameter space. So if you now had some prior idea of what the distribution of this would be, you could put that sort of Bayesian view on top of this.
But isn't the probability of a finite chunk of an R to the power 12, isn't it by definition 0?
It's a finite chunk of interval 0, 1 to the power 12.
I only look at between 0 and 1.
Because I can, if I sample my parameter space from this interval, then I can sample any ratio of parameters. Everything else will only scale the time scale of the dynamics. So will not matter for ideas of stability. Yes. To explain the meaning of these irreducible ecologies, we actually have to prove a theorem. And this theorem requires two definitions, which are, in some sense, just obvious formalizations of the language that I've been using. So I'm going to say that, which is why I've grayed them out, a topology is possible if it allows stable and feasible coexistence for some parameter values, and the topology is an extension of another topology if the second can be obtained from the first by removing one species and all the instant edges. And with this, we have the following statement. Any non-trivial extension of a possible topology is itself possible. This has a very strong consequence, namely that the possibility of coexistence is determined completely by classifying the impossible and irreducible ecologies. Stability and feasibility of a general community emerge from stability and feasibility of small irreducible ecologies. If you want to prove this theorem, actually two cases, depending on whether the added species is growing on its own or whether it's dying. But in both cases, the idea is that if we take the sub-ecology that is possible and add a species, we can make the larger ecology possible. We can find parameter values for which the larger ecology allows coexistence, but choosing the coupling to be sufficiently weak. In the second case, the idea is similar, except that one has to choose an intermediate asymptotic coupling.
But does it mean that I can extend it as many species as I want in the first place if I just couple weakly existence?
Exactly, yes. If you start from an irreducible ecology or from any possible ecology, if you add species sequentially by recoupling, you will find stable and feasible coexistence. So you can build up stable and feasible coexistence in this way.
But then this loses the point of classifying the topology to possible and impossible, no? Because now it's a general mechanism of making any possible, like any ecology possible by just putting weak coupling.
Except for example, the irreducible ones, because for the irreducible ecology, by definition, all the sub-ecologies are either trivial or impossible, so you cannot extend them in this way. So their stability and feasibility does not emerge by this mechanism.
So maybe this is the way that I should interpret that theorem with the minimal sub-ecology. Based on that, you can extend is how to call it?
Is an
keystone species or whatever.
Exactly. So there is a minimal sub-ecology which is an irreducible ecology. I'm not even sure that it's unique. But in general, the possibility of stable and feasible coexistence will emerge then in that way. So to take our results beyond three species ecologies, we need to classify, first of all ecologies of more than three species. This is equivalent to enumerating the complete graphs with 2 colored nodes and 4 colored edges. 4 colors because the point interactions have 2 directions. Fortunately, there's a projection between this problem and the set of directed graphs with self-loops, which can be enumerated efficiently using established tools of computational graph theory. Under this projection, it turns out that non-trivial ecologies correspond to ecologies in which each vertex has positive out degree. And again, those can be enumerated efficiently. And so we obtain ultimately, of course, the expected combinatorial explosion of the number of ecological communities. We have no hope of analyzing numerically all 10 to the 11 ecologies on 7 species, but we can go beyond 3 species. So here is a swarm plot of the probabilities of stable and feasible coexistence for all 2,340 ecologies on 4 species. Of course, by now one has to be a bit careful about how to do the numerics, but these probabilities vary over more than 10 orders of magnitude. They are in fact lower than in the 3-species case, and the irreducible ecologies, again, still have lower probabilities. But the main point to take away from this is that amongst these 2,340 ecologies, there are at most 11 impossible ecologies. I'm saying at most because this is a numerical result. The ideas that we use to prove impossibility for 2 and 3 species ecologies are not sufficient to establish impossibility of these larger networks. We can even go to 5 species ecologies. I'm not plotting the swarm plot now because that would just be a large blob. But amongst these 248,436 non-trivial ecologies on 5 species, at most 30 are impossible. And indeed, if we plot the proportion of impossible or irreducible ecologies against the number of species, it looks as though this proportion is decreasing at least exponentially. So in other words, the possibility of stable and feasible coexistence emerges from an exponentially small fraction of all ecological networks. And this actually brings me to the end of my first example. I've shown you how stable and feasible coexistence is non-trivially impossible in this very small subset of impossible ecologies, and that this the possibility of stable and feasible coexistence emerges from a similarly tiny subset of irreducible ecologies. We can use these results to make more noises about the general trends of different types of interactions on species coexistence. But there's also more mathematics hiding in this question. And in fact, recently, together with some of the mathematicians who are now populating the Center for Systems Biology Dresden, we've been able to map this problem into Grassmannian structures. And these are words that I only learned to use a couple of months ago, and use the power of what is called the Plücker embedding of the Grassmannian to simplify essentially the Rath-Hurwitz conditions of stable and feasible coexistence. There's still much more of this structure to be explored, but this actually allowed us to prove that some of these 4 species networks that we believe to be impossible numerically are actually impossible. More mathematical details available on request. I'm now going to switch gears completely and move to my second example, which is perhaps a bit closer to the main body of our research in theoretical biophysics. This is in collaboration with the group of Star Schwartzman at Princeton, the Flatiron Institute. Experiments were led by his PhD student, Daniel Alber, and the theory was done with my PhD student, Sheng Zhao. I need to introduce first the experimental system that we used in this context. And this is the early development of the fruit fly Drosophila, which is perhaps one of the most well-studied model systems of developmental biology. The embryos of Drosophila are these ellipsoidal sheets of cells, vaguely turd-shaped, that will develop into the fruit fly. Biologists describe these objects in terms of the embryonic axis. The anterior that will develop into the head, opposite the posterior, the dorsal side that will become the back of the fly, opposite the ventral side. Now, the early Drosophila embryo has 2 particular tissues that have been studied to death. They are the germ band and the posterior midgut. The germ band is initially on this lateral side and on the ventral side of the tissue and is genetically patterned to undergo a process of germ band extension. Cell intercalations drive an extension of the germ band around the posterior pole onto the dorsal side. The midgut is genetically patterned to undergo cell shape changes that invaginate the midgut into the ellipsoidal tissue. So in a video, this process looks like this. Here's another view of this process that again shows this germ band that is extending around the posterior pole. The extension of the germ band over the course of only 20 minutes translates the midgut from the posterior pole onto the dorsal side, where it folds into the bulk of the tissue. Now, the germ band and the midgut are separated at this stage by another important tissue, which is called the hindgut primordium. The hindgut also undergoes complex shape changes during morphogenesis, starting as a ring surrounding the posterior pole, and then deforming into this shape on the dorsal side that I'm going to call a keyhole shape. Unlike the germ band and the midgut, the hindgut is not genetically patterned for active deformations. So we were wondering, could this be an example of a different kind of morphogenesis, of passive morphogenesis, driven by forces applied at its boundaries by the surrounding tissues? To test this, we introduced a minimal 2D model that encloses an area A, which is initially A0. Now, the invagination of the midgut reduces the area enclosed by the ring, so reduces, oh, my internet connection is unstable, how good. So reduces A, while the progress of germ band extension determines the positions of the anterior-most and posterior-most points on this ring, so we'll fix a diameter of this ring to some value D. For example, its initial value D0. Now, if there's only midgut invagination, so if only this area A is reduced, but the diameter is not fixed, then this ring will deform into the shape of lowest elastic energy, which is this sort of ellipsoidal-like shape. There are higher modes that have more lobes, but they have higher elastic energy, so are not observed. This is actually a classical problem of elasticity theory. But if we now add germ band extension to this model, so if we add a fixed diameter to this model, then the shapes of lowest energy are asymmetric. There's a fourfold degeneracy of these shapes, but they include the observed keyhole shape. Again, they're also symmetric shapes, but they have high elastic energy, so they're not observed. Up till now, I've asked you to take all of this on trust. So let me perhaps give you some of the details to explain where all of this is coming from. This kind of problem is determined by a Lagrangian that includes the bending energy of the elastic ring, proportional to the curvature squared, the constraint of inextensibility, and the constraint of the fixed enclosed area imposed by a Lagrange multiplier that is essentially the pressure. We can then write down the corresponding Euler-Lagrange equations, which define a boundary value problem with an interesting bifurcation structure. For example, if we fix the diameter to be equal to the initial diameter of the ring, then as the pressure increases, different modes bifurcate off the undeformed branch A is equal to A0. The first mode to bifurcate off is an asymmetric mode corresponding to the asymmetric shapes. This mode later merges back into a symmetric mode that winds back and forth around more asymmetric modes. And if you think that this bifurcation diagram is not complicated enough, then you can ask yourself, what happens if the progress of German extension actually also changes the value of the diameter that is imposed? Well, if we also vary d, then pairs of then these branch points can disappear, or pairs of branch points can merge. I'm only showing this bifurcation diagram because it was really unpleasant to compute. But the meaning of this is seen by now projecting this diagram onto the area and diameter axis. Part of this diagram is geometrically excluded for inextensible deformations of an elastic ring. But in a large chunk of the remaining parameter space, underneath the point corresponding to the initial circular ring, the mode of lowest energy is asymmetric. This means that this symmetry breaking is robust. The model can explain the symmetry breaking of the shape of the Heine-gut, but it cannot yet explain the shape selection. How is this fourfold degeneracy of the mode broken to select one shape? For this, we need to take this model.
Yeah.
Yes.
Two questions. So you talk about asymmetry, but this picture that you showed certainly has asymmetry. But so is there a specific axis that you're talking about when you talk about asymmetry? Or
When I think here I use the word asymmetric to mean that the mode breaks symmetry in the anterior to posterior direction, which is the axis of which the symmetry is broken in the biological problem. Of course, the shape has a symmetry.
Yeah.
Yes.
But then the other question, so you said it's very, the left picture was not very nice to compute, but what kind of tools did you use? Like anything fancy like Auto or two-path or
Auto.
Auto?
Yes. Auto will compute this bifurcation diagram for you actually reasonably nicely, because the equations are sufficiently simple that you actually want to code them up with a Fortran.
And who was crazy enough to do this for you, or are you?
I did not give my PhD student a choice. Unfortunately. Sorry, this is being recorded, I shouldn't say that. But
Okay.
Well, no, this is actually not too bad. I think the only thing that's numerically a bit unpleasant is actually to continue these branch points to the point where they merge. But it's again, that is a two-parameter continuation, so that is actually again doable.
I also have a question about the way of four-fold symmetry, like the previous slide. Do we have this kind of structure depending on the initial condition, like the previous?
Exactly. So these four degenerate modes, or the modes have this fourfold degeneracy because all of these modes have the same elastic energy. What would break the symmetry might be, this degeneracy might be broken, or a mode might be selected by initial conditions, but actually I'm going to show that it is actually broken robustly by geometry, because some initial conditions would not break the symmetry robustly.
So basically, you have a division in the basin of attraction for this four structure.
Exactly, yes. So if you were to select the perturbation of the ring, some of the modes would be preferred. But actually, we remove this degeneracy by the geometry of the embryo. And this was actually the next point I was going to make, that to explain shape selection, we need to take this model from the plane to a curved surface. The same symmetry breaking happens on a curved surface. an elastic ring with a diameter fixed on a spherical surface breaks symmetry into the same four modes as the area enclosed is reduced. And the same thing happens at the posterior pole of an ellipsoid. Again, the modes remain degenerate because of the symmetry of these surfaces. Actually, this is numerically now a bit more unpleasant because the reason why one can solve this problem numerically in auto in the plane is that the equation, the Euler-Lagrange equations can just be expressed in terms of the curvature. On the surface, one has to distinguish between the normal curvature and the geodesic curvature. Both of them appear in the equations. And so you would have to encode the relation in these curvatures, which depends on the embedding, which is a complete nightmare. So rather what we did is to define families of shapes in terms of a few Fourier coefficients in ellipsoidal coordinates, and then we can minimize the energy directly and obtain these shapes. But now, if we take such a ring from the posterior pole of an ellipsoid and move it to the dorsal side, the anisotropy of curvature will break the degeneracy of the modes now robustly, and it will select one shape. And indeed, it does select the orientation of the keyhole seen in the experimental system. So this anisotropic curvature is a mechanism from which shape can emerge robustly.
But then this means that you can create an extra dimension in your phase diagram.
Exactly. So
Exactly.
So geometry gives you this extra dimension that makes shape selection robust. We can say more, in fact, about the kinematics of this process, but biologists would of course not be happy until some perturbations have been introduced. So the way one does this is that here's again the normal fly, the wild type. The model predicts the symmetry breaking that is also seen in the experiment. But we could also look at various mutant flies in which some processes have been abolished. There are flies in which there's no medial invagination. There are flies in which there's no germ band extension because there's no germ band. One can even get rid of another process called ventral flower invagination, which is a bit more complicated. But in all of these cases, the model predicts shape changes consistent with the ones observed in these mutant flies. So both biologists and physicists can be satisfied, hopefully. And I'm getting to the end of the second part of my talk, which is that biologically, this minimal model can explain this process of hindgut morphogenesis in Drosophila as the passive mechanical result of midgut invagination and germ band extension in the surrounding tissues. More physically, perhaps, the geometry of curved surfaces can serve as a mechanism for shape selection. And this actually prompted us to think more abstractly about a related class of mechanical instabilities that happen within curved surfaces. And to answer the question that I guess Euler forgot to solve 200 years ago because he didn't have Mathematica, which is the question, how does an elastic line buckle not in the plane, but how does it buckle if it is confined to a curved surface? The answer is, it depends on the curved surface. But in general, there is beautiful new bifurcation behavior with additional post-buckling instabilities that can be explained by doing lots of asymptotics and also in terms of more general dynamical systems theory. And this is really thanks to the reviewer of this paper who asked really good questions as the unfolding of a perturbed pitchfork bifurcation. I'm now going to move on to the third part, which is part of the PhD thesis of my student my MC today, who actually submitted her PhD thesis 2 days ago. And this is on another problem of tissue mechanics, in particular on a process called epithelial gap closure. That is something that's very common in different developmental systems, where a tissue closes around the egg in development. This process is often associated with a contractile cable that surrounds the tissue and that pulls the gap closed after it has sort of passed the equator in this picture. But if you think about it, to go from sort of this stage to this stage, the length of the boundary has to decrease. And so if the cells are not to deform, not to undergo huge deformations, then there must be cell rearrangements where the number of cells around the gap decreases because cells deintercalate from the gap into the bulk of the tissue. A very well-known example of this is what is called epiphyly in zebrafish. The embryo or blastoderm closes around the yolk, driven by cell divisions that expand the embryo. But a very simple example of this process is what is called serosa closure in the beetle tribolium, because this is an example of epithelial gap closure without cell divisions. The serosa, an extraembryonic tissue, closes around the embryo. And in this video, the boundary between the two tissues has been highlighted by these dots. And why is this not shown? Interesting. This again. So the gap closes, the number of cells surrounding the gap is reduced. And so experimentally, it has been observed that cells deintercalate from the boundary into the bulk of the tissue. It has been suggested that this requires the tissue to effectively fluidize in the vicinity of this gap. And it has been observed that the cable that is contracting the edge of the gap is inhomogeneous, with the cells that have a higher contractile actomyosin activity being squeezed out of the gap into the bulk of the tissue. So these are sort of the observations that we wanted to understand mechanically, but also more generally, we wanted to understand the interplay between the elasticity of such a tissue and the irreversible plastic rearrangements of cell intercalations in this tissue.
Yeah, is there anything special about the cells on the boundary? Is there some genetical marker that is present that makes them separate from the rest, or?
There's nothing special about these cells, except for the presence of this cable that surrounds the boundary, that is essentially defined by the boundary between two different tissues. But otherwise, they're genetically the same. And so, as a minimal description of this kind of process, in the language of nonlinear continuum mechanics, we think of the deformation as a mapping between an undeformed configuration and a deformed configuration. Our minimal model of serosa closure is one of the closure of a circular gap in a flat circular tissue. This deformation will decompose into slow plastic intercalations, followed by fast elastic relaxation. This defines an intermediate configuration after cell intercalations, which is in general not geometrically compatible, because due to cells intercalating, and due to the number of cells in each ring surrounding the gap changing as a consequence, the preferred length of the ring of radius s will change from 2 pi s to 2 pi s times some f of s and t, where f of s and t is an intercalation stretch that determines the geometric incompatibility resulting from these intercalations. Elasticity must then intervene to glue these different rings back together and deform the tissue. The elastic part of this deformation happens on a scale of seconds, much faster than the intercalations themselves. So this will quadrastatically minimize the mechanical energy of the system. This mechanical energy is the sum of an elastic energy of the tissue itself and the contribution from the cable, proportional to the contractility lambda of the cable. The elastic energy can be expressed in terms of the strains in the tissue, and is proportional to an elastic modulus, which is an overall constant that will not matter in our analysis. These strains are defined in these coordinates as essentially the stretches in the different directions and depend on the deformation and on this intercalation stretch. So to close this model, we need to impose the dynamics of the intercalation stretch. So we need to impose the dynamics of these plastic intercalations. For this, we need to start with a microscopic model of cell intercalations in such a tissue, where a cell in a ring of radius Si surrounding the gap can jump to the outer or inner radii with microscopic intercalation rates ρi plus minus. These intercalation rates and the number of cells in each ring then define an effective outward intercalation rate and its dynamics. Because the number of cells in each ring is proportional to this intercalation stretch, we can coarse grain this model to write down the dynamics of this intercalation stretch. The partial derivative of the intercalation stretch with respect to time depends on this intercalation rate. One parameter that appears in this equation, this will be important later, is beta, which is the effective aspect ratios of the individual cells in the model. Now, this model already hides some interesting physics because actually time reversal symmetry is broken at the boundary in this model. The reason is the following. If cells at the inner boundary intercalate into the gap, then the intrinsic radius of the gap must decrease. If cells at the inner boundary intercalate outwards, then they do not remove this inner ring of cells, so the intrinsic radius of the inner ring does not change. And this breaks time reversal symmetry at the boundaries of the tissue. We can now finally close this model by imposing these microscopic intercalation rates and relating the intercalation rates to the mechanics of the system. To do this, we need to think about the energetics of an individual intercalation in some intercalation coordinate. This involves crossing an energy barrier to intercalation at the T1 transition point, at the point where the topological rearrangement happens. Afterwards, this will be accompanied by a release or gain of mechanical energy due to this topological rearrangement, delta E. The intercalation rates will depend on both of these quantities, and the simplest model we can choose is an A-ring model, where these intercalation rates are proportional to the exponential of these energies, divided by some effective temperature theta that measures contractile cortex fluctuations in the tissue that drive these intercalations. The energy released or gained by intercalation delta E is a property of the elasticity and is already prescribed in our model. The energy barrier, however, is something that is a property of the cell mechanics and in general has a complicated dependence on cell shapes, on the state of stress in the system, and we don't know anything about this. So here, we're just going to study two limits of this model. The first is the limit of a high energy barrier, in which this energy barrier is constant and swamps the mechanical energy released or gained. The second limit is that of a fluidized tissue, where fluidization means that the energy barrier vanishes compared to the energy released or gained. In both of these cases, we can crank our algebraic handle and compute the effective intercalation rates. It turns out that the high energy barrier regime is rather boring. In the expression for the effective intercalation rate, the cell aspect ratio, beta, only rescales the effective temperature and the energy barrier. But in the fluidized regime, a term beta squared appears, and this is actually an emergent geometric effect of cell shape on the intercalation rates that has an interesting consequence. Indeed, we can model an inhomogeneous contractile cable by replacing cell aspect ratios, beta with an average value beta, angle bracket, and also replacing beta squared with the average value of beta squared. Now, it turns out that in this expression, the second derivative of energy density with respect to the intercalation stretch is positive, and by convexity, the average of beta squared is less than the average of beta squared. This means that for an inhomogeneous cable, the absolute value of the intercalation rate increases. In other words, an inhomogeneous cable, as observed in the trivolume system that motivated our study, increases intercalation rates, favors gap closure. Moreover, we can now solve the dynamics of the model in both of these cases. First of all, in both cases, we observe that the gap starts to close, the radius of the gap decreases with time. In the fluidized case, actually, it seems that the radius of the gap converges to a constant value. This is biologically not an issue because it's known that in many related systems, the contractility is also an increasing function of time. And so this gap would actually start to close completely if the contractility were to increase with time. The mechanical energy of the tissue at first decreases, but then afterwards starts to increase, which again is not surprising for this out of equilibrium model, in which mechanically unfavorable intercalations can persist due to the breaking of time reversal symmetry. What is more interesting is the fact that the macroscopic dynamics in both of these systems are actually different. In the high energy barrier regime, the intercalation rate is negative near the gap. This means that cells intercalate into the gap to close the gap. In the fluidized regime, the intercalation rate is positive there. Cells deintercalate from the boundary into the bulk to reduce the number of cells in the ring surrounding the gap and thus to close it. These are, of course, the two mechanisms that one could expect to close a gap, but these two mechanisms emerge at the macroscopic scale from the different cell scale energetics of intercalation. In the trivolume system, we know that cells deintercalate from the boundary into the bulk, and so only the fluidized model is compatible with the observed dynamics, which is consistent with tissue fluidization inferred by cell shape analysis previously in this system. So this actually brings me to the end of my third example, in which I've shown you that these opposite macroscopic dynamics can emerge from the microscopic details of the mechanics. There's also the emergent role of cell shape variability, which in homogeneities of such a contractile cable can favor the gap closure process. More generally, we hope that this kind of model can provide a framework to think about this interplay between tissue elasticity and rearrangements, plastic rearrangements of cell intercalations. And so one thing that remains to do in this context, I think, is to extend this framework to describe the full tensorial intercalation dynamics in a bulk 3D tissue. And finally, this is tribolium. Thank you very much.
Some point more semantics. So you call this folding, you call it passive, but in some sense it's also active in the sense, isn't it? That is, I mean, this embryo is actively making these germ bands and these.
Exactly, but I was calling it passive morphogenesis because it's passive morphogenesis of this particular tissue, whose morphogenesis one gets for free from the active morphogenesis of the surrounding tissues. So in some sense, it's an example that shows that the system doesn't need to program all of its tissues to deform actively. Some of them can also undergo complex shape changes from these boundary forces resulting from other tissues.
But it's more like, if you see folding happening in whatever embryo, it doesn't necessarily mean that there is something that this is programmed in the sense that where you see it happening, then it's not necessarily happening there. It might be happening.
It might be happening elsewhere. Yes.
Elsewhere. Yeah.
And I guess you could ask so the more abstract question: what kind of shape deformations can you program in this passive manner? Especially, how are different shape deformations in different tissues compatible with each other? So if you want to, if tissue A is prescribed to deform actively in a certain way, what can you make tissue B do as a consequence?
And I assume that all these genetically modified embryos, they are not viable, or
they are not viable for different reasons, mostly happening later. So, for example, that is mainly because not much has happened by this point. That's the point of studying early morphogenesis in some sense. For example, these BCD Oscar, bicoid Oscar embryos essentially have the way you remove the germ band is to make sure that the embryo has two posteriors, so it doesn't have a head. So it's clearly not going to be viable.
No.
Bicoid Oscar Spätzle, by the way, they are all named after Germans because they have all German names because they were all discovered in Heidelberg. And does not have the ventral furrow formation, so that means that it essentially doesn't have a spine, if I remember correctly. So definitely not viable. Folded gastrulation doesn't have a gut. So something will definitely go wrong.
I have a question. So here you show only 4 different shapes, and you mentioned that is the fourfold symmetry breaking. But does it mean that in your Lagrangian you only have these 4 possible configurations? Because I can imagine that many different shapes would satisfy the being minima of that free energy or whatever. So I'm really surprised that why at the end of the day you get only 4 shapes.
So it is the only the lowest mode that has a fourfold degeneracy. So the degeneracy comes from the fact that there's nothing in this model that breaks symmetry. There is so no symmetry in this direction, which means that I can take this bit and also reflect it. And so then I get this shape, and I can rotate these by 180 degrees, and I get these two shapes. So that symmetry is not broken spontaneously. The higher modes have similar degeneracies.
To what extent are all these higher modes relevant for biology? Is this ever being observed in?
No, because they are they have higher elastic energy, so it would be impossible to realize them essentially. I mean, the way this works in the buckling problem in general is that if you buckle a sheet of paper, it will buckle into the first mode. You can force a system to buckle into a higher mode, essentially by compressing something, by relying on inertia. So you might compress something fast beyond the threshold for the second mode, and then you don't have time to excite the first mode, so you will excite the second mode.
What was your motivation for computing this?
You just let auto run. I mean,
Okay.
that is why we did not compute this for these nodes. But there's actually one more serious point to make. There's another way of removing the higher modes, which is that, and this is something that I did not mention at this stage, which is that this is no longer a true bifurcation, but a continuous deformation, we believe. Because in general, this would be related to the idea of unfolding a pitchfork bifurcation again. As you perturb the pitchfork, it's a continuous deformation. And then the second mode essentially moves even farther away. So it's no longer connected to an undeformed solution.
So, no, then we thank Pierre.
Thank you very much.